Okay, good morning, good afternoon, or good evening, depending on where in the world you are joining us from today. Please do let us know in the chat. I am delighted to welcome you to the 4th IPRRG webinar series, which we have arranged in place of our annual meeting. And remember, if you have any questions during the webinar, please ask those using the Q&A function. So today I would like to welcome Tania Yanov. Tanya is a senior research scientist and describes herself as an accidental ecological modeler. Her undergraduate and PhD studies were in zoology, and fieldwork in Malawi gave her a taste for applied ecology. She undertook training in Australia in modelling using the first version of CLIMEX, and this was the start of a career combining ecology with modelling, of using modelling not only as a management tool, but also as a structural platform to synthesize current knowledge and understanding of pest species, and to help prioritise future research for effective management of pests. Tanya published the first application of climate change scenarios to understand potential pest risks. She has extensive experience in developing climatic niche models to understand pest risk, contributing to the citrus black spot risk assessments and bioeconomic assessments for the Harvest Choice project. She also crafts process-based population dynamic models to better understand how to manage impacts. And her most recent projects have been using pest risk models to understand biological control systems. And it's the topic of biological control systems she'll be speaking on today with her talk entitled Accidental Preemptive Biocontrol for the Brown Marmorated Stinkbug. Thank you, Tanya.
Thanks, Melanie. I will now attempt to share my screen, which I have lost. All right. Slideshow. I can start. Sorry, Melanie, can you remind me? Share screen down here first.
Yep.
This one. And slideshow.
Yep.
We will go play from start. Thank you, everyone, for showing up. All right, excellent. Thank you, everyone, for showing up. As Melanie indicated, I will be talking about, do we have accidental preemptive biocontrol for the brown marmorated stink bug, and looking at different pest risk techniques to assess potential agents. Before I go any further, I need to thank all my collaborators without whom this project would not have been possible, colleagues in Australia, colleagues in New Zealand, and in the USA. Thanks to everyone. Okay, just to put everything in context, as you are probably all very well aware, BMSB is a major pest and is rapidly spreading both through North America and in Europe. And there are two Trissolcus species that are key parasitoids of BMSB in Asia. The first is japonicus, which has very high parasitization rates in the native range, and in lab experiments has also been shown to have very high parasitization rates. And this species has already invaded North America, Italy, and Switzerland. Trissolcus mitsukurii is one of the key parasitoids in Japan, and it has also now invaded Italy. It occurs in Australia, although we are unsure of its actual invasion status. We only have a total of 17 location records for it here, 4 of which predate 1930, the rest of which post-date a specific introduction in 1962, where it was brought in from Japan to control the green vegetable bug, Nezara viridula. We presumed for this entire study that it is actually a naturalized exotic here in Australia, although we have some sampling happening at the moment, and hopefully some population genetics work will happen to work out whether it's invaded or what its status is. Okay, so the main research questions are, given that BMSB is spreading, number one, do the projected invaded ranges of BMSB and those two egg parasitoids overlap? So can they be considered to be inventive biocontrol agents for BMSB where they now occur in North America, Europe? and in Europe. Okay, we know that there are existing CLIMEX Compare Location models. Criticos et al. Did one in 2017 for BMSB, and Avila and Charles in 2018 for japonicus. Our problem is that there is virtually nothing known about Trissolcus mitsukurii. There are simply 2 papers published with any biological information whatsoever. So now we're at the problem of, if we want to look at where this species can occur, do we do a match climates or do we do a compare locations? Those are basically where we can go. Match climates is a very simple and straightforward process. It's quick, it's easy, you need no biological information. All you're doing is simply assessing the similarity of the climate in an area where you know your species occurs to a climate in your area of interest. The problem is that because there's no biological information, you can end up with problematical comparisons. Not scope to go into all of that there. We can go into that later if necessary. Compare locations is a lot more complex, a lot more time consuming. You actually have to have some knowledge of your species biology, and you really need to have some understanding of basic ecological principles so that you can select plausible mechanisms that will limit a species distribution. You can't just throw numbers into this thing and say, oh, it works. However, it does use different types of data sets, whether it's lab experiments, whether it's phenological data that you have from different locations, and you can look at seasonality and population growth, and of course, location records that give you different kinds of information on the climate at these locations. As a result, you do end up with a much more robust analysis. Drawback is, it's not as easy. Before I go any further, regardless of the type of analysis that you want to conduct, you really need to understand what a location record means. Do you have an interception record? Do you have an established population? With both of the Trissolcus location records, this is actually fairly easy. I think we had a total of 60-odd for mitsukurii and 100-odd location records for japonicus. So it's fairly easy to work out, yeah, okay, they're probably okay. For BMSB, this was not a minor undertaking. There were hundreds of thousands of records in GBIF. And when you map these, you're going, does it really occur in England, or are they interception records? Does it really occur in Switzerland? Or sorry, not Switzerland, Sweden. We know it doesn't occur in New Zealand, but you've got records there. So before you even begin your analysis, you can't just download location record and say, okay, I'm just going to throw this in the analysis and come up with, a result. You really need to understand what data you're using. Okay, so from our perspective, we decided that since we had minimal data on the biology of mitsukurii, that we would start with the match climates analysis and try to see what other areas in the world have a climate similar to that in its native range. Location records, what we did is we actually separated these into specifically native, which is the records in Asia, versus exotic, i.e. in Italy and Australia. We also then wanted to consider rainfall, agriculture versus irrigated. We know that mitsukurii is a host of BMSB, which is an agricultural pest, therefore likely to occur in areas because agriculture is practiced with irrigation. This is a little bit different in that we specifically separated native versus exotic locations. Most of these sorts of analyses take all location records and then just match all known location records with away locations to find out where else is suitable. We also decided that, well, since we can do a little bit of CLIMEX modeling, we'll also try the reverse type analysis where we can look at, well, how do the climates in the exotic locations match those of in the native location? Okay, because we were interested in looking at the impact of irrigation, the factors that we used are only 3. Your standard match climates within the CLIMEX framework is minimum temperature, maximum temperature, rainfall total, and rainfall amount. But because we wanted to consider the effect of irrigation, we can do that if we use soil moisture. You add irrigation, obviously soil moisture increases. And okay, we use the standard CLIMEX 10-minute grid for our metadata. Methodology that we used is, as I indicated, we started off with our native point locations, and we intersected that with the global map of irrigated areas to work out which areas undergo irrigation and which don't. It gave us 25 native locations where irrigation is practiced and 7 where it is not. When we did our climate match to the rest of the world, we were looking to see the rest of the world under irrigation or under natural rainfall. So in both cases, you have your native locations, you do your climate match against irrigated or not irrigated away locations. For irrigations, for locations that fall within irrigated areas, that fall within the GMIA and therefore are irrigated, we wanted the match in the highest possible match index. So we took the maximum match index from all of these results to get that one. The reason or the rationale behind that is that, well, we don't really know when irrigation is applied. We don't know whether it's applied to crops where BMSB occurs or whether it occurs at some other time. So we're just interested in the best possible match. For the locations that fall outside of, the irrigated areas, we simply took the best possible match index from locations where there was no rainfall applied, and we produced a composite map where we basically mapped the irrigated areas with the best and the non-irrigated areas with the best. Then we specifically looked at how that analysis matched, so what the match index was for exotic locations, both Australia and Italy. Italy had a low match, more concerned at this point with what happened in Australia. As you can see, there's actually a relatively low match. The match index is 0.55, which on a scale of 0 to 1 is not real flash. In addition, there are 2 location records you can see there where they're being excluded by this analysis. In other words, the climates in those 2 locations do not match anything in the native range, which would imply that you should not find the species here. The climate's very different, so you would not expect it. So if you were going out looking for a biocontrol agent, you wouldn't be looking, to think that it could survive here. Then we thought, well, okay, that didn't work very well. Let's try the standard analysis, where we can no longer now consider irrigation. So we've gone back and reverted to the standard 4, which includes rainfall amount and rainfall pattern. As you can see, that also ended up excluding a very different point, but that point is now excluded because it's exceedingly dry here. Agriculture only occurs with irrigation, so not surprising. But again, the take-home message is it's not working terribly, terribly well, and you're still ending up with a relatively low match index here to try to include your location records. Most papers and most other studies that have used this type of analysis have indicated that with the CMI of 0.7, that's sort of the standard when you want to make these kinds of comparisons. So we're going, we're still not real happy with this. Just because we can, we did the reverse climate match where we took location records in Italy and Australia, matched the climates of those locations, and looked to see what happens in Asia. And as you can see, again, we have a matched climate index of 0.6 is necessary to include all of the location records in Asia. So again, you're left with the, well, it works, sort of, but it's not real flash, which is what we know pretty much about this kind of analysis. If you put them all together, you end up with these three. So this is our original where we did soil moisture and looked at Asia versus the world. This is the 4-factor where we instead used rainfall amount and pattern against the world. And this is our reverse where we looked at comparing Australia and Italy to the rest of the world. Take-home message is all the maps are somewhat different, basically. So again, highlighting that you're not getting a great result here. In all cases, to include location records, your match index is fairly low, ranging from 0.55 to 0.65. But then again, you can also say, well, okay, on the other hand, they're also somewhat showing the same thing. There's lots of differences. There's also quite a few similarities. So the key message being, in all cases, mitsukurii should be able to spread well beyond its current range in Asia. In Europe, one would imagine that if it's already established there, all of these analyses show that it should be able to spread further. So lots of differences in the projections, which gives you limited confidence in the whole methodology. Nonetheless, it implies that mitsukurii should be able to spread globally. Okay, we didn't get very far with that analysis, so we decided, where can we go? And the only option is basically to do a compare locations analysis, even given that we have minimal data, and then to see if we can come up with something and consider the range overlaps of all three species. So next step in the analysis was to do a compare locations. We generated a model for Trissolcus mitsukurii. Again, with our location records, what we did was we used the native locations in Asia to parameterize, train the model if you like, and all exotic locations to validate the model. So these exotic locations were not included when we were setting the parameter values for CLIMEX. Same meteorological data was used, and we produced same sorts of composite maps. We actually considered rainfall and irrigation. With the CLIMEX, you can do that. So the final composite map, we ran the model. Once we had our parameters, we ran the model with rainfall or not. And then if locations fell within the GMIA and are irrigated, we used the maximum EI value to show suitability. And this is what we came up with, which all in all isn't too bad. The nice thing is that all exotic location points, so all of us, every single point in Australia and in Italy fell within regions that were predicted to be suitable. So having parameterized it, we didn't actually have to go back and change anything. What this shows, same as all the match climates analyses, is that there are a lot more areas in the world that are projected to be suitable for the establishment of this species. And the nice thing is that we parameterized the model, ran it. So our exotic locations fell in, but we had two new location records that we discovered after we did the analysis, the one in Iran and actually this one out here at Charters Towers that we discovered afterwards. Both of those fit in, so we went, okay, we're reasonably comfortable with the model. When you look at what do the different kinds of analyses tell you, effectively, you're left with, well, there's lots of other parts of the world that mitsukurii can spread to where it doesn't currently exist. So in a sense, our compare locations model, the final one, is a bit of a compromise between sort of here where you can get, massive spread everywhere, and here you get much less spread. It's a bit of a compromise. But generally, similar areas are projected to be suitable. So there's a concurrence between the results of those two types of analysis. But it does point out that you do have to be very careful when you do the match climates, when you have limited numbers of data points, when you're looking at just the native range versus everywhere else. You actually probably have more confidence in a model where you've included some biology. Probably what this points out is that we need to also assess the, as it continues to spread, look at where mitsukurii does end up to maybe be able to assess the climatic tolerances that it can tolerate. That may not be being captured by the limited range that we have in Asia. Given that they're all here, we know that there must be, it must occur here. It's just a sampling. We just don't have the sampling. It's an absence of location points. It doesn't mean the species doesn't occur. Both BMSB and japonicus occur down here. It's highly likely that Trissolcus occurs here too. Therefore, you know, you could end up with a better projection. So we did what we aimed to do, and we can now look at the projected distribution of all species. So we've got H. halys, japonicus, and mitsukurii, and our location records that we have for all of them. And in general, you could say that, sure, there's differences in them. Japonicus can go further north, but in general, the two Trissolcus species should be able to expand and cover the range of BMSB wherever it spreads to. So that means that in the USA, where japonicus already exists, there's no reason that it can't spread now to effectively end up covering the same territory as BMSB. Similarly, in Europe, japonicus is already there, as is mitsukurii, so both of them should be able to spread there. In Australia, we have mitsukurii, so hopefully it's already here, and if when BMSB arrives here, we already have it here to control. New Zealand is an interesting point. They are very concerned about BMSB arriving in their country, and they have actually already pre-approved the release of japonicus. So if BMSB arrives in New Zealand, japonicus will be released. It can, both japonicus and H. halys can occur in New Zealand. That's not an issue, so hopefully they will control it. It is also likely that if both BMSB and japonicus end up in New Zealand, that we'll also end up with japonicus, even though we will not specifically import it, we're probably likely to end up with it. So we may end up with another adventive mitigation factor. Since probably many of you are in Europe, I thought I might zoom into Europe a little bit and show you what happens more closely in Europe. Again, the maps of the three species with slightly different projected ranges. But again, the main take home is that your two Trissolcus species should be able to occupy the same space. Now, before you get too bent out of shape about the fact that, oh, BMSB should not be occurring here, BMSB does occur there. So this is the projection for BMSB. And this takes me back to one of my earlier slides saying, what is in a location record? So yes, the existing CLIMEX model that was developed in 2017 is this projection. Now, as you can see, most of Switzerland is projected as unsuitable. That does not mean that the model is wrong. Stokely et al. Actually used exactly the same CLIMEX model, but ran on a much finer scale metadata. Their metadata was on a 2-kilometer grid, not the 10-minute grid. And they came up with a series of maps because they did it over various years and then looked at the impacts of climate change. And in their top map is what CLIMEX predicts using the metadata for that particular year. So this is the 2018 year and 2018 weather data. And you can see that they sampled BMSB in areas where CLIMEX predicted it to be suitable. They never got it in areas where it's unsuitable. Similarly, in 2019, the model predicted slightly different areas. So climate obviously varies and changes. And nonetheless, where they sampled is where the model said that BMSB should occur. However, if you look at the two, I mean, these are just two out of their multiple graphs. You have, for example, these sets of locations that are recorded as obviously location records. You have BMSB occurring in several locations here and sort of all down this area of Switzerland, which you don't get in the 2019. However, there's those, these are the ones that go across the bottom, which are all of these points. However, you know, are these on either side really permanent location records? Apparently not. So that doesn't mean that the model's wrong. It means seasonality. It means a whole heap, you know, annual differences in temperature. So the fact that CLIMEX here has not predicted suitability doesn't mean that the model's wrong. It means, okay, you really do need to look at these data points and see what is happening. On that basis, I sort of discount much of what happens in Germany and Austria. Same idea. It's all highly topographically variable, which the scale of the CLIMON data set will not capture. More interesting is what happens in the Netherlands. We have 10 location records, which actually fall in areas that are projected to be unsuitable by that CLIMEX model. The reason they do so is that the model says there are insufficient degree days for a generation to be completed. In other words, you can't complete a generation, you can't lay your eggs, you can't keep the cycle going. Each of these locations is a single individual that was taken. So this is downloaded from the GBIF records. Again, what does a location record mean? We have single adults. There was one nymph. I don't recall precisely which location it was in. So the one nymph means, okay, you had breeding. All the others are on buildings and in built up areas. Does this mean that these are actually interceptions and nothing happened? They were single individuals. Does it mean that you actually have populations that now can persist because we're heating up buildings, providing effectively warmer microclimates where BMSB can now hide and potentially complete a generation? We don't know this. Nonetheless, when you use a model and you then drill down into the, you know, what you're seeing, it's really clear that you have to know what your location records mean. From my perspective, I'm not in Europe, but I would suggest that in the Netherlands, they actually go and look. If BMSB might be a pest, then they might want to know how, why, or do something about it. Okay, and on a similar note, zooming into Italy and Switzerland, on the left are two maps that have very recently come out by Zapponi et al. And the survey was done in 2019. The top left is for Trissolcus japonicus. They had some initial data sets and then they just went out looking. The red and white dots are where they sampled and they found BMSB. On the right is the japonicus model run and the original location data. So some of these occurring are the ones that Zapponi et al. Have used as well. So for example, we've got two records in Switzerland from Stahl et al. We have a Sabatini Peberieri data point here and one from Moravio et al. What this shows is that all of the areas where they have now found japonicus are actually being projected to be suitable by our model. So we didn't have the data points, but the model's looking pretty good, especially when you start looking, you know, you've got it here, all that's being projected as suitable. There's a valley up here. Lower down for mitsukurii, it does not occur in Switzerland yet, but it has definitely spread throughout Italy, northern Italy. This whole area here, again, all being projected as being suitable, including they've got these bits where they've sampled mitsukurii heading up in this valley towards the border. We only had the one sample, but the model clearly shows that these areas are suitable. Whether you end up exactly at this location point and then it becomes a case of scale and what metadata you're using, not sure. However, our analysis do confirm that both of these species will be able to spread and hopefully will be able to help mitigate the effects of BMSB in Europe. Before I finish, I have to acknowledge that there were lots of sources of BMSB data. Many colleagues provided information, and obviously I geocoded some as well from the literature. Similarly for japonicus, many colleagues provided their own records or what wasn't available as well as literature. Very little for mitsukurii anywhere, but it is what it is. Thank you to everyone. Final point in terms of all of this is that all of the location records for these species that I've collated for this analysis, I will make available on the IPRRG website. They'll be available as spreadsheets or CSV files if you want to create your own shape files, and there will be as reformatted shape files that you can simply use. It will be possible to update any of this information with new information. So for example, if we get access to the location records in that Zapponi et al. Study that has just come out, we can add them there. Ideally, you would want to know that they really are permanent established species as opposed to it's a seasonal incursion, but that is, you know, it is what it is. But that information will be made available and anyone who's interested can have access to it. Thank you very much.
Thank you very much. So remember, if you do have any questions, you can use the question and answer function to ask those and I will pass them on. But perhaps I could just kick off. You can stop sharing your screen if you want as well, Tanya.
Sharing your screen, stop sharing. There we go.
There we go. So I thought the detail you're going into about sort of the individual records was a really important point to make and really interesting because it's something that comes up a lot when we're sort of doing risk analysis as well. What is a real record? Do you find it difficult sometimes to, you know, how do you go about sort of qualifying these individual records and do you find it easier or is it sometimes quite challenging to figure out what might be an established population or what might be just an incursion, just a single interception?
It varies. In the case of the Trissolcus species, I pretty much took it as, okay, they had been sampled. Now with mitsukurii, some of these samples are old, we don't really know. So with mitsukurii, I am treating them with a grain of caution, but it is all we have. Mm-hmm. The detail I went to with BMSB was when I first downloaded the GBIF data and then mapped it in QGIS, I'm going, I know that New Zealand does not have it. So why is it showing up here? Well, hang on a moment. I really didn't think that it could go as far north as Sweden. So I contacted people in these countries. So I mean, one of my colleagues on the paper is in New Zealand. Yeah, no, it really does not occur in New Zealand. It's an incursion later. It's part of the problem that we now face, I think, more and more with citizen science. So as I pointed out, even published records, so that Stokely et al. Paper is fabulous, but when you start looking at it, you know, are they really established populations? No, they're not. So many of those, I couldn't sift through hundreds of thousands of GBIF records. To some extent, you have to decide, well, okay, for what I'm doing, the bulk of them are falling within what I think is okay space. You know, you have BMSB in Switzerland. Am I really going to get bent out of shape over a couple points? No. It can be explained for a variety of reasons. But I do think it's very important to address those points, same as the points I addressed in the Netherlands. I don't study BMSB over there. I don't really know much about BMSB. Does it occur in the Netherlands? All I've got are these data points. They've been put there by someone. Citizen science, probably. The pictures are on GBIF. Okay, they were found. Are they really, that's then where you use the model and say, well, it should not be able to exist there. I have confidence in that model because the model is saying there are insufficient degree days, and it wasn't just a figure plucked out of the sun. They actually went and they looked at a range. I mean, there must have been half a dozen references. There were some reviews done. The model is based on data. So I trust the model. Now you go, well, okay, so what do those location records mean? And that means sending it back out to whomever to say, well, okay, go find out. Is it real? Are we heating the world up and BMSB can now occur here and then they overwinter and then they become a problem in your fields come spring? Or are these really incursion records that, okay, yes, they're GBIF, but if you're going to do any modeling or if you're going to look at them, you need to know something about the biology.
Mm. And I imagine that's quite you were saying that you've got very little data for mitsukurii. And I was just wondering, is it also a species that's perhaps very difficult to identify? So perhaps it's actually much more widespread than we know. It's just people don't know how to find it or look for it.
That is probably very, very true. So, I mean, we had 29 location records in all of Asia. We know it has to be wider spread than that. I didn't hone in and do a zoom in, but certainly in some of the match climates analysis that we did, you're looking at where BMSB occurs and then looking at where japonicus occurs, and they're in this whole province of southern China where there's not a single record there.
Yeah.
Given that they overlap elsewhere and that there's no geographical barrier, there's nothing to stop it being there. So what we have is, yes, a paucity, an absence of records. It's not that the species doesn't occur, it's just that we don't know where it occurs. People really haven't been bothered to look at it until very recently because BMSB has become a pest. So until you get that information, you're really largely stuffed.
Yes, I think I think you'll probably have triggered interest from quite a few people in going out and looking for those too. Especially, I noticed that it spread quite almost beyond the range on some of the maps of where you'd expect brown marmorated stinkbug. But like you said, the other thing I found really interesting was the sort of the zoomed in records. Which I think are really important when you were showing sort of Switzerland and how perhaps it looks like there's a big gap, but when you zoom in, you can really see that you've got these sort of individual areas. So I was just thinking, sort of how important do you think it is to be looking at that very fine degree scale to figure out where things may be able to establish?
If you're in the UK, for example, and you're asking us, okay, as a management in the UK, because you've had a number of incursions there already, but as far as you know, they are only incursions. If you really want to know where it will go, I think you trust the biology of the model. If you really want to go further down and say, well, I mean, you haven't, as far as you know, you haven't had it yet. Do you need to go to that level of detail and go to fine scale metadata? Until you have it, probably not, because the model is telling you that it should not be able to survive there. I think I reviewed a paper very recently of some colleague, actually, I think with DEFRA, sent it to me. And, they were saying, well, okay, there's a handful of locations that might be suitable. They might be suitable, but you don't have it there yet. So until you have it, it's pointless. Really. So you can use the model as is to say, okay, we don't think it will go. You will also get possible annual variability. So, I think we showed this in another model that we did way back when in Africa. You'll get annual variations in climate. You can run CLIMEX on that to see, is this going to be a good year? Or, is this a good year compared to previous years? So if you do end up then with a small population somewhere, is that population going to continue or not? Well, what's your average versus, okay, that year was a particularly warm year. If you look at the Stokely et al. Paper, obviously the 4 years that they sampled, they have very different maps. I just picked 2 of them to show here and to zoom in to say, okay, you can quickly see some differences in their location records. Each year is very different. And each year, maybe some years are favorable, maybe some aren't.
Okay, so we've got to hope for cooler summers in the UK to continue, actually.
Yes.
So they have been very warm in recent years. So I think that might actually be, let's just double check. You've had some very positive comments on the chat, but I think we've actually covered all of the questions now. So I just want to thank you very much again for presenting today. And thank you everybody for attending, and please do go onto the IPRRG website and check out our upcoming webinars for May and June. Okay, thank you very much everybody. Bye bye for now.